Okay, so today we are going to look at the ABC superfamily of protein pumps. Now, why are these called ABC superfamily? Such a fancy name. So, oops, just make it black. So the reason they are called ABC is it's an acronym for ATP binding cassette. As you can see, the ATP, which means it will bind to ATP and use the hydrolysis to generate energy, makes it a this makes it an active transporter, and it's a protein pump which is using energy. Now let us look at its structure before we go into its functional details. So it has two transmembrane domains, and each these are called the TMD, transmembrane domain, and they usually consist of 6 alpha helices. The usual notion, but sometimes they can have 5 alpha helices, or sometimes they go to 10 to 12 in some superfamily proteins. Now the function of these alpha helices is to provide specificity for their substrate, and also they are the passageway, they are the tunnel through which the substrate moves. Now look how it functions later. Before going to that, let us look at how the energy for this activity comes from two ATP binding domains, and these are called the NBD because here the N is nucleotide, because ATP is a nucleotide. These are called NBD, nucleotide binding domain. Now these domains are necessary for dimerization when a transportation is to take place. Usually this ABC superfamily can be present as single unit also, only one transmembrane domain and one ATP binding domain, but they cannot do transport of uh, substrate across membrane. They can only relay signal inside the cell at max, but having two ATP binding domain gives them the ability to become a transporter. So as I have already mentioned, they can form single monomeric unit also, but they can form dimers, and this dimer can be homodimer, or it can also be heterodimer, giving them the flexibility to um, work with different kind of. One point to note about these is, because of this variability, they are the largest family of transmembrane protein, and their family can be recognized through the presence of some of these conserved ATP binding domain, and some of the motifs present in this NBD, which we'll look soon. So these are responsible for the transport of small molecules, like not ions, rather small uncharged molecules or some basic polar molecules. They can also transport ions, but they mostly transport amino acids or sugar molecules, and even drugs, like the medicines we take. Once they are metabolized, they can be transported through this kind of ABC superfamily proteins. So as I was talking about the different motifs present in the NBD, there are two important motifs called Walker A, or Walker A domain, or like I think it's better to call it motif, which is the one responsible for interacting with the nucleotide or the ATP. So the Walker A motifs interacts with the nucleotide phosphate group, and the other one, the other motif, as you would have guessed from the Walker A, the other one is called Walker B. And the Walker B motif is responsible, or rather it is helping in the catalytic process by binding or interacting with the uh, Mg divalent magnesium ion, and also with water, so it makes a coordinate bonds with them in the catalytic side. which is finally providing the catalytic basis for the nucleotide hydrolysis. So now we can look at how it actually functions by looking at an example. Here the example is the P-glycoprotein, also called ABCB1, which is responsible for some of the drug transport across the membranes. And here you can see this is one TMD1 and TMD2 and NBD1 and NBD2. And each TMD has 6 alpha helices which are spanning the membrane, and the NBD are present towards the intracellular side. Now, as I was talking, there is a Walker A motif and there is a Walker B motif in each NBD. Now, along with this Walker A and Walker B, there are other motifs called H loop, Q motif, and such different motifs with different amino acid composition. But Walker A and Walker B are the most important ones, so I've only mentioned those two here. Now, as we have seen a little bit about the structure, now let us look how it actually functions. So here you see there are two like the TMD1 and TMD2 are present to make it like a tunnel, and the NBD are towards the intracellular side, and there is ATP present inside the cell, and then the substrate here is present inside towards the intracellular side. So what happens is once the substrate binds and ATP binds, it forms a closed structure, and it in a way sends the substrate towards the extracellular end, and then it assumes a structure which is opens towards the extracellular side, and it releases the substrate from inside of the cell to the outside of the cell. Now this kind of motion can happen in the opposite direction also. Once the transport has happened, the ATP, the bound ATP in the NBD, it hydrolyzes into ADP and inorganic phosphate. That opens this, that again brings back the structure to how it was before the hydrolysis. And we'll look into this structure soon enough in a molecular detail. But before that, we can see why this is really so important. There are a lot of these kind of ABC superfamily protein, and some of the, as I have already mentioned, this is the largest superfamily or largest family of transmembrane proteins, and a lot of them are transporting mundane only amino acids or sugars, and they are present in both prokaryotes and eukaryotes. But even being so diverse and there being so many members, they can be sometimes clinically really relevant. So I'll just put some medically relevant examples here. So the first one I'm going to talk became popular because it has a role in cancer treatment, or rather resistance of cancer to drugs. It is called MDR1, multi-drug resistant 1. It's found in mammals or mammalian cells, and what it does is any cancerous cell which overexpress this MDR1 can gain drug resistance, because MDR1 is responsible for the efflux of drugs. So cancer cells try to overexpress it and cause more efflux of any drug that is given. So now it is becoming a popular target for cancer therapeutics. Now, other than this, another one is called the CFTR, and the CFTR, the CF stands for cystic fibrosis. I think you might have heard about this disease already. So I'll just write it here, cystic fibrosis. So this is actually a chlorine ion transporter, and it transport it across the epithelia of our epithelia. And what happens is when there is a dysregulation of this CFTR, then there can be cystic fibrosis. So these were two medically relevant examples. Now let us look at the structure. Okay, here we have the P glycoprotein, or also called ABCP1 according to the ABC transporter nomenclature, and this in short is also called PGP for P glycoprotein. Now this protein is from human with the structure I am showing here, and you can see that it is in a dimeric form with a closed state where the two NBD domains are close to each other, and you can also see the TMD domains with their 6 alpha helices here. And I can try to zoom in the NBD where the ATP binds, and you can see the stick figures of the ATP molecules. So as it is bound to ATP, it's in a closed state and the NBDs are close to each other. Here we have the same protein in its open state. As you can see, the NBD domains are away from each other. This is because it is captured after the hydrolysis. It is crystallized after the ATP hydrolysis has happened. And here, if you remember the figure I had shown you, it is in the orientation where the substrate can enter from the inside of the cell. This is the extracellular and intracellular, and this is the transmembrane domain, and the substrate can enter from the bottom and then move to the outward side.